Dr. Hans is medical doctor, associate professor, CBO of Cell Receptor, a small Geneva-based research company dealing with a new bottleneck mechanism in cancer cell growth. Dr. Hans studied medicine in Tübingen and his doctorate at the Max Planck Institute for Virus Research, where he worked in the lab of tumor virology. In 1973, he held an IARC fellowship at the Wallenberg Lab in Uppsala, I believe in Sweden. He specialized in laboratory medicine. He received the Vinja Legendi in Freiburg in 1982 for making the discovery of a T-cell growth factor, which later was named interleukin-2. In 1991, he came to the Am Urban Clinic in Berlin, Germany, heading the central lab. From 1996 to 1999, he was medical director of this clinic, and from 2001, he designed and directed the Central Lab Institute for the Viventis Healthcare Network. Since 2010, he has been engaged again in basic biology of tumor cells, focusing on regulatory mechanisms of cell growth. His work has led to 13 granted patents. Wow. So, ladies and gentlemen, you are in store for a treat, and I'll give the stage up to you, Doctor.
Thank you very much for the introduction. I will share my screen with you now and talk on basis of my, let's see, where are we? That is fine. So the Gordian knot is a metaphor for an unsolved, difficult problem. I will talk about the problems in developing and implementing simple and effective solutions in cancer treatment in Europe. Such problems have some different reasons, and I will try to list the most important ones and then present an example. I will start with a look at the pharmacological industry. 20 years ago, a Harvard Business Review about the healthcare industry in the United States was published by Christensen, Boehmer, and Kennedy. This study was very critical regarding the ability of the healthcare industry to be innovative. This situation is understandable and not typical for the United States only. Innovations have an early first phase in which there is no return on the investment made for them. Shareholder satisfaction is very important for the industry. Disruptive developments usually will lead to fundamental changes in the routine. This may cause substantial disruptions also of the cash flow. Such developments are a risk and therefore not very popular in industry. But the future lies in the change. Another problem lies in the medicinal treatment of cancer patients. Let me have a look at this. Important processes outside of the cancer cell are rarely targeted. Medicinal oncology attacks the dividing tumor cell with cytostatic drugs, monoclonal antibodies, checkpoint blockers, all of which are designed to disrupt the vital functions of cancer cells. These agents may have a variety of adverse effects on the patients. Normal cells, especially growing normal cells and cells of the central nervous system, are frequently also affected. Such treatment is a true being between Scylla and Charybdis. Targeted cancer can frequently remedy such disturbing measures. It becomes resistant to the applied drugs. Therefore, to prevent resistance, other anti-tumor agents are added to the first one. The burden on the patient becomes very high. The high rate of adverse side effects limits the possibilities of combination treatments. In cases of upcoming resistance, second or third line treatments may have to be applied. These are frequently very expensive. This is an increasingly troublesome issue for cost carriers, which therefore may be restrictive regarding reimbursements. Despite all the progress in oncology, one of the most important problems still prevails. Metastases cause 90% of cancer deaths. With our present treatments, we seem unable to get a good and cost-efficient solution for this problem. Altogether, we see suboptimal treatment results and frequent intolerances in cancer patients and a heavy financial burden on cost carriers and society. This burden causes pressure on care and pricing and promotes relocations of production out of Europe and an unwillingness regarding developments, a Gordian knot. Now, having envisaged industry, clinic, and society, let me look into research. We have Gordian knots there also. I will choose a potentially disruptive project that we started some years ago. Let me start with a glimpse into cell cultures. This is an excerpt from the NCI Register of Cell Cultures. You are looking at 5 human cell lines here in the left column. All of them have mutations of the CRAS gene, a so-called proto-oncogene, and are therefore considered to be self-sufficient in their growth signaling that is supposed to cause the cancer. If you look at the 6th column, you will see the culture medium in which the cells are thriving. And the information in the 7th column is today of very high importance. The cell needs some calf serum in the culture medium to grow. If we would try to keep the cell in a culture medium alone without calf serum, it wouldn't grow at all. Essentially, every cell line in culture needs such a supplement to the culture medium. Therefore, essentially no cell line is self-sufficient in its growth signaling. But such self-sufficiency has been a dogma in basic research and has sadly led nowhere. A terrible Gordian knot. Let us have a look at why cell lines in culture love serum. The serum contains a lot of important substances, hormones, proteins, trace elements, and lipids belong to these. However, just one category of substances is of crucial importance for the initiation and maintenance of cell growth. These are growth factors, substances that are specialized for this function. How are these substances getting into the serum? Let us have a look at a cellular component of the blood. These are platelets. Quite to the right are normal platelets, and at the left lower side you see platelets that have a sort of legs or tails. These are activated platelets. Metabolically active platelets may start blood clotting, a very important function of them. Blood clotting is started by compounds which the platelets transport in granules. Please have a look at the rightmost platelet on the image. This is an electron microscopic image of such a platelet. It carries a lot of dark spots. These are the granules we see in the rightmost platelet. There are different types of them. One of them is the alpha granules, which are containing everything a platelet needs for blood clotting. Alpha granules also carry other substances, growth factors. These are produced in the bone marrow in the precursor cells of platelets, the megakaryocytes, and then passed on to the platelets for transportation in the blood. Platelets are our most important transport vehicles for growth factors. The process of preparation of serum activates platelets. They release growth factors into the serum. That is why cells love serum so much. Growth-committed cells in our body must be able to provide themselves with growth factors. We have no serum in our blood. Otherwise, they would not be able to divide. Let us have a look at how cells do this. If we look at the image, we look at normal human connective tissue cells, fibroblasts, which are components of our connective tissues all over. These cells are young and growing in culture, and you see a lot of small black dots on them. These dots are platelets which we have put into the cell culture just for a few minutes and then washed away again. So you see, the cells have the ability just to bind such platelets that are passing by. The second image shows you a colony of cancer cells. Here you see the same phenomenon. The cells are covered by platelets. It is indeed possible to cultivate cells in the complete absence of serum if you regularly provide platelets to the cells. The next image is a strongly enlarged tumor cell. At the left, you see an activated platelet sitting on the cell with one long protrusion as you saw them before. In the middle part of the tumor cell, you see another platelet that has had somewhat longer contact with the tumor cell already. It has opened up and emptied its contents into the cytoplasm of the tumor cell. Platelets do not contain only growth factors, they contain the molecular machinery that is necessary to produce these factors also. In basic research, it has become clear that platelets and tumors are forming an unholy alliance, resulting in tumor growth. The chain of events that I have demonstrated to you is a part of a mechanism that our body utilizes to control cell growth. This mechanism has not yet been targeted in oncology. Today's oncological treatment is directed against the metabolism of the growing cell. I have already mentioned the drawbacks which are related to this. Metastasis is caused by tumor cells that are shed into the blood system. Only a tiny fraction of circulating tumor cells will end up forming metastases. In the blood, the tumor cell will immediately acquire a clothing of platelets. The platelets help the circulating tumor cell not only to withstand shear stress and attacks by the immune system of the body, but also to form metastases, usually in some distant organ. The platelet cloak is therefore essential for the tumor cell. Circulating tumor cells without the platelet cloak have repeatedly been demonstrated to perish. Let me show an effect in cell culture. The image shows cancer cells covered by platelets as you saw them before, and the right image shows the same experiment. Here, not only platelets were added to the cells, but a substance that is known to interfere with the substance of platelets. This substance prevents platelets from sticking to the tumor cell. The substance we used in the experiment is an anticoagulant, a preventer of blood clotting. It is a low molecular weight heparin. Close relatives to these heparins, highly sulfated saccharides and highly sulfated heparan sulfates, are also able to do this same effect, and it happens at concentrations that are interesting for clinical treatment also. Let us have a look at this in an animal system. This is a publication from last year by the team of Lubo Borsig and Mauro Pavao, headed by Cristiane Silva. Mice were getting tumor cells inoculated into them, and a part of the mice also got a heparan sulfate, which had been isolated from the black sea squirt, a very highly sulfated substance. At the left side, you see the sacrificed lungs of animals that did not get this substance, only saline as a control. And you see that these lungs are full of metastases. Animals, on the other hand, that did get this heparan sulfate did not show any metastasizing into the lungs, and they did not show any take of the tumors either. So what about human beings? Can we use that in humans? Here is one example. Altogether, there have been many thousand patients incorporated in studies looking at the effect of heparins on the growth of tumor cells. This is a study that Mustafa Altinbas and his team in Turkey made in 2004. The solid line in the left graph represents patients with an advanced lung cancer that got low molecular weight heparins in this small time period, just a few weeks before the start of the study. The study lasted almost 3 years. And you see that the patients which had got the short treatment, they had altogether a somewhat better prognosis. They needed longer until the tumor started growing again. And if Altinbas and his team took patients with a limited disease only, you see that this difference is much better. So there was something happening in this time of the treatment that had a very long-lasting effect on the patients. And this will quite certainly be the vulnerability of the circulating tumor cells in the cancer patients. So that is known. All these studies made on such patients, they have had very unlucky study designs. Nobody came on the idea that circulating tumor cells might be a solution for a better treatment. Let me show you one patient. We have had only one such experience so far. This is a lady living not far from our home, 67 years old. She had lost 15 kilograms in 9 months and got abdominal pains. She went to the doctor. A gastric cancer of signet ring cell type was diagnosed, a very unfortunate type of gastric cancer that very early makes metastasis and very early kills the patients. She had already an invasion into the large bowel. She had an abdominal dropsy with numerous signet rings shown in the microscope. She had liver metastases, lung metastases, and enlarged lymph nodes in her chest. She had also suspect osteoblastic metastases in her lumbar spines. She got a conventional treatment. Oncologists know that the treatment in such a stage will have no really convincing result. And last November, this treatment started. It was finished in March this year. She got also as an adjuvant treatment a highly sulfated saccharide, which is approved by the FDA and the EMA. It is given once weekly under the skin of the belly. A final radiological control in March 2021 showed that the large bowel was free of tumor, no pulmonary metastasis anymore, no enlarged lymph nodes, no liver metastases, no abdominal dropsy, only the change in the spine was not observed, so the metastasis might be there still today. The patient is still today receiving this highly sulfated saccharide. She's feeling well, and she gets other treatment also with the hope that we one day might be able to take out the residues of her tumor and give her a normal life afterwards. So we are quite convinced that we should continue our project. It shows promising results. But there is one important point to talk about, that is the finances. This image shows you the region in which our patents on this treatment are valid in Europe, United States, Japan, Australia, and Canada. We would take into considerations the current treatment costs for this substance, a treatment duration of 12 months, and if we multiply this cost with the number of metastatic cancer deaths in this region here, we arrive at a market value of $10 billion US. This is quite a sum. And it is quite certainly worthwhile thinking more about this project. We will need a phase 2 clinical study before we can go into the market, but it sounds very promising. So that is the end of my presentation. I will change windows back again and give you the screen again. So that is my presentation. I thank you for listening, and I hope you could enjoy it.
Thank you so much, Dr. Hans, for your presentation. If there's any questions for Dr. Hans in the room, please type it over here. I'll give everyone a minute.